We start now with the final, one of the final days of the week. And let me see. Okay, we will start today with the lecture from Antonio Lagares Jr. Hi, Antonio. I have some people here that wants to say hello to you.
Hi everybody.
Oh, I'm going to turn the music off.
It's nice background music.
Sorry. Okay. So, all set then. Today, we are here with Dr. Antonio Lagares, who is joining us via Zoom. He's right now in Marburg, Germany. Antonio Lagares is a staff scientist from CONICET at the Universidad de Quilmes, where he did also his PhD. But right now, he's doing a postdoc in Universität Marburg. And today he's going to talk about KEGG analysis for multiomics. So thank you, Antonio, for being here today with us. And it's a pleasure for us to hear you. So all yours.
Okay, so can you hear me well? It's fine?
Yes, perfectly.
Okay, that's great. So just let me know if you can hear me well. I can try to fix mic or, yeah. So first, I would like to thank Nico and Herman for this kind invitation to participate in the course. It's a great pleasure for me to be here, to be honest. It's quite sad that I couldn't make it to Rosario this year, but yeah, the situation was not quite well here. So yeah, I will try to give my best online, and just feel free to raise your hands if you have any questions, and maybe Herman can just transfer your questions to me. I have to say I don't come right from the field of proteomics. I'm most I would say an end user. I don't belong to proteomics service. I also I'm not doing research in proteomics. It's in the field of proteomics, but I have been analyzing data from proteomics, transcriptomics, and other omics together. So I will try to share with you some basic tools which might be useful for you in order to visualize your data, and just to try to take as most as possible from them. So I will start sharing my screen. Can you see the screen now?
Yes, we are seeing everything perfectly.
Okay, so the talk is titled Hands-on KEGG Mapping of Omics Data. And what I will show you today, it can be applied for proteomics data, but you can also use it for other omics. I mean, if you have quantitative data that you want to map on metabolic pathways maps, you can just follow more or less the same workflow, and it will still be possible to analyze your data. So, the main idea is that sometimes we have tables of data which we can map over pathway maps that are really available and free to use, and it can in turn be quite useful to use these resources, because sometimes there are some features, some connections that between your hits that are not easily understood if you just carry out enrichment analysis and so. And when you map your data over these base maps, you can really find uncover new relationships and new interesting features that may still be hidden there. So the idea is quite easy. So normally we start with this data tables, and we want to go towards the mapping and visualization of our data. And so to do so, we need to to carry out some annotation of our data and then some little formatting to make these tables compatible with the input that we have to use for the mapping software. So since we will mainly, I will mainly show you some base maps which are originated from the KEGG database. I thought about making a very brief introduction to the KEGG database. I don't know if you are all familiar with this database. I will just give you some few insights at the very beginning of the talk, just for you to more or less know what it is about. And then I will show you a simple way to generate your own database for annotations, to carry out your own annotations on your own data. Sometimes you can find these databases available to download for free, and sometimes they are not available or they are even not updated. So it's not that hard to make your own tables. And then after we get our annotations, we will annotate our own dataset. And finally, I will show you some softwares, freely available software that you can use to map these results on the pathways. So to start, so what is the KEGG database? So KEGG stands for Kyoto Encyclopedia of Genes and Genomes, and I think the name now it's a little bit outdated because it's not just that. When it was launched in 1995, more or less, it has been growing quite a lot. But the idea and what makes KEGG quite particular is that it not only has entries of genomes and genes, but it also provides maps, metabolic maps and connections for biological systems, which are very nicely generated, and they are manually curated, and they are very trustable. So in general, so there are wiring diagrams of all cellular components, how they are connected with each other. So if there are reactions connecting cellular components, or just structural connections, interactions between different cellular components, everything which is known, the current knowledge, it's taken into account, and this is what they call the systems information. And this is kind of species independent. So there is like a basic layer in which every connection that is known to take place in the cell tries to be integrated into the database. And for that, we will find different kind of chemical information. So small compounds, enzymes, reactions, everything connecting cellular components is integrated into this systems information. And in addition, we have genome information, which comes from sequenced genomes. And from these genomes, we have genes, and the genes that are annotated in the genomes, some of them have functional annotations as well. And these functional annotations are mapped over these base maps of cellular metabolism. So just to show you how it is structured. So basically, there are 4 categories at the moment. I will just mention the main ones just for this talk. So this systems information, which summarizes everything what is happening in the cell at the metabolic level. It's depicted in these KEGG pathways. So this database of pathways are networks of cellular components that we know that they interact with each other. Some of them are the canonical pathways that we learn in biochemistry from intermediate metabolism, but there are some others which are not actually pathways, but they are, in the broad sense, they belong to metabolism, like structure of translational machinery, transcriptional machinery. So everything tries to be integrated into the database. Then, as I told you, compounds reaction classes, biochemical reactions, enzymes, everything is connected with everything and depicted in these pathway maps. And then we have this genomic information in which genomes from different organisms are annotated, and of course the corresponding genes. And if the genes have a function which is already known, so they are grouped into orthologous group of genes, and these groups are coded in this KO database. So each KO object in this database means that there's a group of genes which share the same function. At least they are supposed to have the same function. So every database has its own type of objects, and for instance, the pathway database have maps, which are called with the prefix map, and the prefix can change, and you will see the same number with either with map or with the organism prefix, and that means that we can have the base map, which just describes how the cellular components interact with each other. But we can also, if we have information, genomic information for some specific organism, then we can plot this onto this map, and we will see which reactions are actually taking place in this in every single organism of interest. So functional orthologs grouped in KO numbers, genomes with their corresponding code. Every gene in the genome, regardless that they have or not an annotated function, they will be called after the organism code and then a specific number for the gene. And we can also find all cellular small molecules named after a C, like a compound, reaction after an R, and so on. So it's quite huge. And just to show you an example, we can see this kind of base maps. I will try to. Do you see the Explorer now, or? I don't know if it's switching from the PowerPoint.
We are seeing your PowerPoint.
Okay, so now just 1 second, I think. I will share the whole screen. So can you see now the Explorer?
Yes. We are seeing the Explorer right now.
Okay, so each pathway looks like this, and you have different compounds, which are these dots, which are connected with reactions. So you see here the last code is for the reaction. Then we have in the previous the enzyme code, and then the KO codes for those group of orthologs that are known to catalyze this reaction. And we can switch from this generic map, for instance, to the map which belongs to Saccharomyces, for instance, for the budding yeast. And you will see that now it shows those genes which are predicted to be present in this genome. They are colored in green. So yeah, you can play around with your own organism, and you will see which features are present and which are not. So I will move to the next. So what makes the KEGG very interesting is that it also allows from the very beginning, when it was launched, it offered a KEGG mapper software, which is meant to allow the user to interact with the database with its own data. So we have quantitative data that we generated with an omic approach, and then we can map our data to the database directly with this mapper that the database itself offers. And this mapper has been updated several times. Actually, the last update was this year, and there are several mapping tools. just to mention, if you have a new genome sequence and you want to annotate, reconstruct the pathways that are predicted to be present in your organism, you can just run this reconstruct software, and it will show you which pathways are predicted to be taking place in your organism. There are many other options, but so this color tool, I would say, is the one that I will show you today how to use. And it's just that when you have your codes and you have your quantitative values, then you can upload them into the KEGG maps and see how they look like over the maps. So to start with this presentation, let's say, I will, of course, we need a table of data. And so I chose one data set of study, which we used last year in a different course. And I think it's pretty nice just to use it to explain its features. So it comes from, I will just mention, it's a proteomic analysis of some budding yeast strains, which are natural resistant to plant dry mass, to some inhibitors that are present in plant dry mass. And so they analyze the proteomic response of these strains to the presence of this kind of stress, to identify which proteins were actually taking place in this stress response. And they performed the classical analysis of the data, and so after filtering for statistical significant genes, they found a group of genes which were responsible for this behavior of stress tolerance in both strains, or either in one or the other strain. And they also carried out some enrichment analysis for some Go terms. So they found that there were some biological processes or metabolic functions that were enriched in those genes that were up or down regulated. And they also use some kind of softwares which are also available for free to map these genes to the pathways. And as you can see here, for instance, this paper, they show these maps in which every single object here in this square in the right side, on the right bottom side, it's one of the genes that were found to be either down or upregulated upon this stress, and they colored them according to the function of the genes, and then it's quite easy to see which functions are enriched or not in this response. But they didn't go further with this kind of analysis, and so I will try to show you how with this data we can map how can we map this data over the pathways to see what we can see in addition. So before I start, so as I told you before, sometimes we can find these annotations files quite easy for our organism of interest, and sometimes they are not really available. And even if they are available, sometimes I found some inconsistencies, or the layout of the files are not you cannot properly work with them directly. So I would recommend, if you don't find your annotation files, you can just download it and make it your own. So you can always have a look for instance here at the Perseus server, and for many of them you will find annotations, and some of them are quite outdated. Maybe 2 or 3 years ago they were uploaded. But if you want to create your own database, it's quite easy, and I will leave you here the steps, but I will show you very briefly how to make this annotation table of your own. So I've seen that there is a new version of the UniProt, so I will just try to use it so that you here. So let's try to generate our own file for the budding list. So we need, normally when we analyze our proteomics data, we use the UniProt codes, and with these codes we cannot directly go to the KEGG mapper because they won't be recognized. In former versions of the mapper, it was possible to upload different kind of codes, but the new versions of the mapper just need you to load the KEGG symbols for every hit that you have in order to understand them and be available to map them over the maps. So how can we cross link our UniProt codes with the KEGG codes? It's quite easy. So you can just search for your organism of interest. So it will show you. Let me just. I haven't done this before, and I think now I'm not here. So you first have to go to the proteomes, here, and then you search for your organism of interest. So you are on the UniProt database. Within the UniProt database, you go to the proteome database, and then you search for your own strain. Let's say this one, ATCC strain of the budding yeast, and you click here, and then you will see all the replicants listed here, and you can just go to view the proteins. So then you see all the hits which belong to this organism, and it will normally show you by default some columns, and you can customize these columns. And what we will do is we will just simplify it. We will get rid of those that are not of our interest now, and just leave. So now we have the UniProt code, some protein names, and we will add, by coming here to these external links, you can go and search for genome annotations. And here you have different kind of annotations, and KEGG is here. So we add this column here. And so we have a new column here, which is the KEGG cross-reference. Once you have this table, you just download it as a tab separated values, uncompressed, so there's no need for compression. And just download all tab separated values, uncompressed, and just download. And this will generate a file. So this could be really from Saccharomyces cerevisiae, and we can just check this file. And you just have the different columns that you were seeing in the web page, just in this file. So this file is the one that we will use for annotate our own data. Sometimes UniProt doesn't have all the codes that you need. So sometimes it's not enough. So if you're working with a different code system, which is not a UniProt, and you want to cross your entries with different with different kind of nomenclatures, you can also use different servers to cross these entries, type of entries. And there's, in my case, for instance, we normally use the locus tag, which is a different system for naming the genes. And it's quite useful, at least in bacteria. Mostly we use this kind of nomenclature. And this locus tag is not easily accessible from the UniProt. So there are also other servers. I would say DAVID is one of those, which is quite useful, with which you can enter the UniProt codes, and you can get as output for instance, the local stats. So you can play around with these servers and try to get your the entries that you need just using as input the uniprot tags. So then you can merge all the tables and get your final annotation table that you will use for annotate your own results. So I will also leave here some steps that I will share with you. So in case you need to use this database, you can just follow these easy steps to create your own files. So I guess you have been working with the data, with this kind of data and maybe annotating it. So I will just go, I won't stay quite longer at this step, but so once we have our table for annotations, we need to annotate our data. And just let me know if you're familiar with this. I will just go faster, but maybe there are some people which are not taking part of the course, and just I can show you how to annotate your data. I don't know if it's okay or...
Antonio, we are using the same data set that you are showing us, but we are not doing this part. So I think your lecture is covering completely and closing the circle perfectly with what we are doing, because we are using the data set to teach the students Max Quan and Perseus. We arrived to the volcano plot. So this part is great that you can show us how to carry on with visualization with KEGG. So great. We are using the same data set.
Okay, so it makes sense to annotate the data, or you already...
Have you annotated the data? Yes, the instructors show how to annotate the data in Perseus.
Okay, okay. So once you annotate the data in Perseus, then you will have... I will... just a moment. I think I will have to do it anyway, because I think I missed this intermediate file, so I will just do it very quickly. So we have our uniprot codes, our fold changes, and I will just annotate yeah. So after you annotate your data, you will have a table. What I've done is I filtered the tables that you already know, just to have here for one single experiment those hits which were found to be either up or down regulated in this experiment. So here you see a table with 120 hit items, and all these genes are genes that were either found to be down or up regulated. So this is something that you have to do in advance before you start working with this data to map it into the pathways. So once you have this table in which you filtered all those hits which were differentially accumulated in your system of interest, then you can go move forward to the next step. So so now we have our data tables annotated, and we want to format them to make them compatible with the visualization software. So which softwares I recommend to do this? So I would go with these first two, which belong to I would say quite huge institutions who keep them updated, and they are accessible at any time. There's also one that I found was published last year, I think, which also allows you to map your data, but it has been published and it's not supported by any institution. So I will show you more or less how to format your data and which are the advantages and disadvantages of both these softwares to map the data. And So I will start with the KEGG mapper, which is the one which I introduced you before, and it's offered by the KEGG itself. And so the KEGG mapper has different tools, and the one that I will show you now is the color tool. It was updated this year, and up to last year, until last year, it was a little bit, I mean, there was an option which was quite nice, which is not available anymore, in which you could, you were able to upload your codes, your gene codes, and your M values, so the fold changes or any quantitative data associated to each of your hits. And this software automatically calculated a color which was connected, I mean, linked with the value. So, for instance, for upregulated genes, it was calculating how light or how dark the green color was to map these genes over the map, and so and the same for the downregulated genes. So this was quite useful, but it is not available anymore. So what we have now is a basic, let's say a basic version of this color tool, in which you can upload your hits along with some color information that you have to specify in advance. So it's not possible anymore to use the M values directly to upload here into the mapper. So we have to modify our data to make it compatible with this entry. So how should our data look like to make it compatible with this software? So we need our KEGG IDs, and then we need to specify the color code for each hit, which has to be in this hexadecimal code. And of course, you can personally and manually give a code for each hit, which might not be the same. I mean, you can either use this code, which would be the green code for every hit which is upregulated, and the red code, for instance, for every hit which is downregulated. But you can also make like a gradient if you know how to transform M values into colors, which is not difficult. I won't go into this detail now because it takes some time. But you can, of course, do this, and you can just map your results with the corresponding colors, which will be according to the actual M values for each hit. But to make it quite easy and to show you an example of how this mapper works, I will just color every upregulated gene with green and downregulated gene with red. So to do so, you can use any software of your preference. It's not that we have to use Perseus, but I find, since we are working with this Perseus to analyze our data, why not to just do some more processing and make this table ready to be uploaded into this software. So how can we modify our data to make it compatible? So we don't need this UniProt IDs anymore once we have this codes here. So the first thing I will do is I will remove the UniProt IDs from the column. So now we have a matrix in which we just have the name and the M value. And then I want to assign a color for the positive values and a different color for the negative values. So here we can just use this filter rows option based on numerical column, and we can tell the Perseus to filter all those hits that have a value over 0, and we won't reduce the matrix. We will just add a new categorical column here. So now we have one column in which all positive values will be kept and negative values discarded. And then what we just have to do is just to modify this column, like a text string column. So to be able to edit this column in Perseus, we needed first to change the type because it's a categorical column. It cannot be edited. So I will change this column. I will make it text. I assume you already know how to deal with these transformations. So now we have this column, which is the text type column. I will also rename it. Rename it, so it will be the color. And then I will just use process text column. And so this color column, we will change all those rows in which we have a keep, which was the value over 0. We will tell the we will change this keep for the color code for the green value. So the green value in this coding system is 0 for red, FF for green, and 0 again for the last color. So this is the code for green. So now you see we have every single hit which was upregulated has its corresponding code here in this column. And I will so process the next column. And so those which are discarded, I will change them by, I will make them be colored as red. So red is FF0000. So now we have, I think, oh, I forgot to add this here. So now we have all our hits here with the corresponding codes, and yeah, they just have the right coloring code. So we don't need this fold change column anymore. So we will just remove this column from the table, and now we have a table which looks pretty nice, and it's what we actually need to upload to the KEGG mapper. So we can go here to save, and we just generate a file with this table. And you just have to go to the mapper. Here to the Can you still hear me? Or... Yeah. Okay, so now you come to this mapper website, and you just have to upload this file, KEGG mapper, here. And we will tell this software that we are working with the padding list. So if you don't know the code, you can just get it from here. In this case, it's FCE. And I will just let every feature as it was set by default. And then when you execute this, you will get a list of those pathways to which some of your hits are mapping. So you can see there are different pathways in which we have a number of hits mapping. And if you enter to those specific pathways, then you will see your genes color in red in this case because they were downregulated. And so this is what I told you, that it's not always like a metabolic pathway that it's depicted in a pathway map, but you can also have kind of structure of molecular complexes in the membrane complex in this case, or the respiratory chain, and you also can see the those hits mapped here in this. And if you put the mouse, if you place the mouse over the hit, you will get some information about this, and you can also click here and go and have a closer look to what is it actually, what's the function, and and it will link you with many other databases like PDV, or so. It's quite nice to just interact with a database like this. And so I will come back to this pathway, and you remember I showed you in the when I introduced the paper that there were some graphs here which they published. Sorry. Yeah, so they carried out this enrichment analysis, and then they mapped their findings in this kind of maps. And there were many hits which belong to purine metabolism and amino acid metabolism. So, and as you can see, if we go back to this KEGG mapper and we take a closer look here, for instance, if we enter to purine metabolism, then you will see how our hits map. And it's very nice to see that it's not only that we know that we have plenty of hits mapping to this pathway, but now we can actually see that they are quite connected with each other. They actually belong to the metabolic pathway for EMP biosynthesis, and all of them are upregulated in this case. So this is quite useful information that you can easily access and see with this software. Also, they mentioned some changes in the pentose phosphate pathway, and so here you can see that there are also many hits in red here in this central part of the pathway. And what is also interesting is that if you also have some data coming from a complementary approach like metabolomics or so, you can also map this data into the same map. And I just make an example here. You can come to this option here, color option, enter here, and you place the codes for the metabolites, and you can give them again a color according to the M value that you found in your metabolomics approach, for instance. And then you execute and you load them, and you can also get colors over the dots for the compounds. So it makes the image more even more comprehensive. So I would say this is what you can do very easily with the KEGG mapper tool. Unfortunately, if you want to have a gradient, a color gradient for the hits, you need to modify the codes in a way that they are not full green or full red. You can just make the gradient yourself. This is possible, completely possible, and the software understands these codes, and it will show you this gradient as well. But it takes a little bit more of time, and I won't go into this now. What I will show you just coming to the end. So with this same data, you can also use the IPath3, which is it's provided by the MEMBL. So it's also a nice software which is freely available. I would say the drawback, the disadvantage of this one is that you cannot filter by smaller pathways. You just have to map your results onto the whole metabolism maps, and sometimes it's not quite easy to understand. Quite easy to understand what you are observing if you have the whole metabolism map pictured there. But just to show you very briefly, so what we need when we want to use this IPath, So what we need with the iPath is, again, a table with the KEGG entries with the color codes, which we already have. But what you can do in advance and very easily is to give every hit a value indicating the width of the lines, if they are enzymes or proteins associated with the reaction, or the radius of the dots if they are compounds, if you have some metabolomics data, for example. So it requires that you format your data in this way, so you have to add this W before the value, and then just the value. And you can use the M value, or you can just multiply it by any factor if you want to make the lines wider, or this is something that which you can also play a little bit, but I will just show you that it's also quite easy to do with this KEGG. So with Perseus, for instance, so with this previous matrix, not with the last one in which we got rid of the M values, but if you go to this previous matrix, you see that we still have the M values here, and what we want to do now is to make all of them positive. So we will transform these values of the M values. We will just calculate the absolute value for all of them. So now we just have the same numbers, all positive, but we still have information of their behavior, because we still have this coloring scheme, and then we just have to modify this in a way that we can upload it with the W. So since it's a numeric, we have numeric type, we will change it again to text, so that we can edit this column. So now we have this as a text, and we will process this text column, so the whole change column. And probably you already used this regular expression here, which tells the software to take the whole content of the cell and save it in a variable that we will recall now. And so we want this expression to be exchanged for the W, and then the same value that was taken from the cell. So with this code, we tell the software to just add the W before the content of the cell. So you will see now we have this process data here. KEGG codes, color values, and then some quantitative data to upload. So we just save this here. So again, really easy. Okay, and then we just go to the mapping software. We will use just as an example, this whole metabolism map. And then you come here to the controls, show advanced options. You upload this file that we just generated iPad. And you can make the background a little bit fainter so that it doesn't interfere with your coloring scheme. So this would be the color of every feature in the map which is not mapped with your data, and then just submit data. See, and then you can see again how your hits map to the different pathways. So here the pencils phosphate pathway again with red lines indicating that it was up down regulated, and here the nucleotide purine metabolism with green lines indicating that the pathway is upregulated, and so you can play around a little bit with these values. You can maybe make them a little bit larger so that you get wider lines in some cases, but yeah, basically this is what you can do, and you can also yeah, navigate into this picture and see which is each gene. And I think sometimes there are connections which are not easily understandable if you don't map them into this kind of figures, because maybe they look to be somewhere else in the metabolism, and then when you map them here over this base map, actually they might be closer than expected. So I think it's a very nice tool as well. So let's go back. Yeah, I think that's, yeah, I will leave 5 minutes for any questions if you have, but I think it's quite a straightforward way to map your own results with very simple formatting steps and annotation steps. And yeah, I hope it will be also useful for you to analyze your own data. So thanks for your attention.
Thank you so much, Antonio, for this great presentation. As I told you in the middle of the presentation, you closed perfectly the circle in what we are teaching here with Max Juan and Perseus. Also, the students play around a little bit with Cytoscape, but we haven't touched upon KEGG and iPad, and so it is great what you just show us. And also, we didn't tell you that we were going to use the same data set, so we communicated telepathically. And it is great that you showed the same data set that, well, we taught last year in another course. So great. I will start with a couple of questions of my own. I guess all of these tools have export options for high definition images. Am I right?
Mm-hmm. Yeah, that's right. Yeah, you can export PNG files or so from the maps. Yeah.
And it was a little small to see in the screen. Can you give us again the regex expression for adding the W? I think it was W $1, if I'm correctly.
Yeah. For this last step, you mean?
Yes. Which was the regex expression that you used there? W $1, I think it was.
Yeah. So process text. Yeah. Here.
Yeah, in the replacement string.
Yeah. So here it's W or whatever you want to introduce into your string,
Great.
and then the pesos sign
the dollars one. Great. That's it. Great.
Yeah. That means that it will take what it has found with this regular expression and it will use it. Yeah, it will just paste it, concatenate it after this W. You can just do it the other way around and it will be, yeah.
Okay. Great. Do we have some question in the audience for Antonio? Well, or maybe someone in the audience in Zoom, maybe? We have here a question from the audience here in the auditorium.
Okay.
It's on? Okay. Hi, Antonio. Great talk. I was thinking about signaling pathways. Is it possible to visualize them with this tool?
Yeah, not with this iPad, I guess. I think they are not here, available here. No, but in the KEGG for sure. So you can play around a lot with KEGG. You will find that, yeah. So, so you have metabolism, genetic information processing, which you can find like this kind of maps for polymerases and every complex in the cell, and then signal transduction here, eukaryote, prokaryote. I mean, it's been constantly updated. Maybe it doesn't have everything which is known, currently known, but there are many new features that I found last month. So yeah, it's. I think you will find mostly what you're looking for. Yeah.
Okay, if they are enriched on the samples, they will appear on the list.
Yeah,
Okay.
absolutely. If you are entering a gene, which I will. So if your gene has a code which maps to, for instance, let's say OR here, which has its own code, you will for sure find this hit there, and it will show you the color that you indicated for this hit as well. Yeah.
Okay, perfect. Thanks.
Yeah.
Yeah, I think we have a question from the people in Zoom. Federico Pascuti is asking, it is possible to use this tool with an unannotated genome, or at least non-annotated in UniProt?
The KEGG, so what you can do, you can first try to find out which KO you have in your genome, which is not annotated. So you first have to annotate it yourself. I mean, you can still use this database without having your genome annotated in the database. So if you, let's say you have a new species or a new strain that you sequenced and you have a genome, you can map I mean, you can annotate the genes that are present in your specific strain, and you can assign for every single gene, if there is any KO group, I mean, orthologo groups which map, which is corresponding to these genes, you can map them with this mapper as well. So you can annotate your genome with the KO IDs. And once you have these KO IDs, what you have to do is just create your own database, generate your own file, like table, and then you just map it with the KO values, not with the, I mean, I will go back here, maybe it's a little easier. Yeah. So let's say you have your genome and you annotate every gene there and you blast to the KEGG, and then it will retrieve you which KOs, which functional ortholog groups correspond to each of your genes, if there are any. So you will know for each of your genes to which KO they map. And then you just can use this nomenclature, this ID, to annotate your table, and then map the KEGG, use this table with the KEGG mapper, with this KO. So it won't be a species-specific symbol, but it will be a KO, which is representative for all genes which belong to the same function. So it will also map this to the metabolic maps. I don't know if I was clear enough.
Great, thank you. Do we have another question for Antonio? Oh. Okay, thank you, Antonio. This was great talk. Thank you so much for joining us from there in Germany. Please, another round of applause for Antonio.
Thank you.
So, thank you, Antonio. We'll see you later.
See you later. Yeah, see you then.
Bye.
Bye-bye.
Okay, great. We are going to take just 5 minutes break to prepare everything for Veronica's talk. So we will carry on with the rest of the morning in a few minutes. Thank you.